Welcome to Episode 7 of Epigenetics Explained. Today I will show you what could go wrong in our epigenome for cancer to develop. So, how do epigenetic mutations, or epimutations, lead to cancer? One possibility is that tumor suppressor genes become silenced. This is somewhat self-explanatory by their name, but they are genes which regulate cell division, remove errors in DNA, or promote apoptosis, which is programmed cell death. These genes are important as their repression is associated with uncontrolled cellular growth and proliferation, all of which are hallmarks of cancer. Another possibility is that oncogenes become activated. Oncogenes are the mutated version of proto-oncogenes, which normally help cells grow. Oncogenes cause the uncontrollable growth of cells, which in many cases leads to cancer. A third possibility is that genes which code for epigenetic regulators, for example DNMT3A, become mutated. Now let's use our knowledge of epigenetic mechanisms such as DNA methylation and histone modifications to understand how their malfunctioning can lead to cancer. So, global hypomethylation is a common feature in cancer. This is associated with increased activation of genes across the genome, since, as you know, hypomethylation is the lack of methylation, and methylation usually means repression. Indeed, 5-methylcytosine content, which remember is the methylated form of cytosine, decreased by an average of 10% in certain cancers. Well, how does global hypomethylation translate to cancer? First, oncogenes could become activated, which, as I said before, promotes uncontrolled cell growth and tumorigenesis, which is the formation of tumors. MicroRNAs, which remember are associated with repression, could be activated as well. This may result in aberrant silencing of proteins produced by tumor suppressor genes. How does a genome become globally hypomethylated, and not just in certain loci or certain locations in the genome? This can occur through disruption of DNMT1, which is the maintenance methyltransferase. Resulting in hypomethylation. Another common feature of cancer is site-specific hypomethylation, which is associated with excess inactivation. This can take place on tumor suppressor genes. Like I said before, they regulate processes which counteract that of cancer. Thus, their silencing promotes growth and proliferation of potentially harmful cells, which leads to tumorigenesis. Site-specific hypomethylation can also target transcription factors. This consequently inactivates their target genes, some of which may be tumor suppressor genes. DNA repair genes are also targeted. Their hypomethylation allows abnormalities to accumulate, catalyzing the progression of cancer. Site-specific hypomethylation can be caused by an increase in DNMT1 methylation, errors in de novo methylation, which is carried out by DNMT3A and DNMT3B, and errors in attempting to remove aberrant DNA methylation by means of demethylation. An example of hypermethylation is that of the gene P16, which regulates the cell cycle. This can be found in colorectal cancers as well as other cancers. Histone deacetylation, which is characterized by the overexpression of histone deacetylases, causes histones to wrap DNA more tightly as opposing charges are restored. As a result, the target gene or genes are repressed. These target genes could include tumor suppressor genes, the consequences of which I have already outlined. Changes in histone methylation patterns can also lead to aberrant gene silencing. For example, the improper regulation of histone methyltransferases, which are responsible for the even distribution of repressive marks, could result in the silencing of tumor suppressor genes. A correlation has been found between the overexpression of EZH2, which is a histone methyltransferase, and the occurrence of melanomas, lymphomas, prostate, and breast cancers. As I've briefly mentioned before, the deregulation of microRNAs also plays a role in the progression of cancers, as they normally regulate genes involved in apoptosis, cell proliferation, and transcriptional regulation, which can lead to tumorigenesis. For example, the downregulation, which is a lack of regulation of microRNA 15 and microRNA 16, results in the activation of the BCL2 oncogene. This can lead to chronic lymphocytic leukemia, or CLL. Thank you for watching this video about the epigenetic changes that could take place to cause cancer, ranging from global hypermethylation to changes in histone modifications and deregulation of microRNAs. It won't be all doom and gloom forever, as our 8th and final episode of Epigenetics Explained will be about epigenetic drugs and therapy. Thank you for coming this far, and see you in the next video.